My name is Rachel Murphy. I am the volunteer manager of the National Plant Monitoring Scheme, and I'm joined today by Sarah Shuttleworth, who's going to give you a big wave. Sarah is a specialist advisor, botanical advisor here at Plant Life, and it actually is very familiar with the National Plant Monitoring Scheme, having worked within the team for about 18 months, wasn't it, Sarah, a couple of years ago. So this webinar forms part of our annual engagement programme. We're running a number of lunchtime webinar sessions throughout the year, along, of course, with our online interactive events and our in-field events. So please do take a look at those. So that's it from me. I'm not going to say anything else. I'm going to hand over to Sarah to make a start on today's session. Thank you, Sarah.
Thanks, Rachel. Brilliant. It's lovely to come back and do these talks for NPMS. As NPMS is pretty I'm pretty fond of it, having worked in it, as Rachel was saying. So today's session, without further ado, so plant anatomy, you might initially think, oh, that's a bit dry and a bit stuffy, but actually we're going to try and make it as engaging as possible. But also it's so important because I don't know if you've ever picked up one of your lovely field guides that you can get these days, and you start to read the descriptive text about the plants and how to tell them apart from each other, and you'll suddenly realise there's a whole load of other words there that maybe you're not familiar with, and suddenly it can just seem a little bit bamboozling. So we're going to try and help with that element, and it will really provide a solid foundation for plant identification going forward. So without further ado, let's make a start. So as I mentioned, so we're going to try and learn the basic parts of a plant, starting off with the sort of floral part of it, which is obviously usually the best thing to use to identify a plant. And then, we'll go into the other parts of the plant as well. But we're not going to go into high levels of detail about vegetative ID, because that's something that, you probably want to do much later on in your sort of plant ID skills sessions. And it's pretty difficult. Even I try and avoid that if I can. We're hoping to be able to then recognise some of these parts and identify them in some example flowers from pictures. And I'd really recommend that you then go out in the field and try and sort of recognise them in live with real plants. So that'll just really sort of build on that knowledge. And it'll hopefully provide this really strong foundation for learning more about plant ID, but also it's often some of this language is used for other things. So grasses, sedges and rushes obviously have their own terminology, but there's also some things that are repeated. So it should be good. And the reason I said for learning this is that it will help you understand how to identify species and what sort of features might be useful to really understand and know the differences between them. And I'll try and cover that with each one. So as I mentioned, there's a lot of bamboozling terminology that we're going to cover. And I've written the list of names of the words here. The ones in bold might be ones that you've definitely come across before and are very familiar with. Some of them may be familiar with, but also just to have a little bit of a trouble remembering which bit is what, especially because obviously stamen, style and stigma can be quite difficult, seeing as they will start very similarly sounding words. But there might be other words on here that are completely new to you. But these are all words that I've taken basically looking at maybe some of the common guidebooks like the Rose's Wildflower Key and the Collins book. These are all words that are used regularly throughout those books, so it will be useful to understand them. So as I mentioned, we're going to start from that top part of the plant that is the flower, and we're going to start from the base of the flower, working up and into the centre of it. Sort of it always starts from the outside in when you're learning about floral anatomy. But leading up to that, you've obviously got your main stem of the plant, but in particular, the sort of little stalk that has the flower, especially with those species that maybe have multiple flowers coming off little stalks, that stalk that holds the flower or the inflorescence is often referred to as the pedicel. And that's really useful. And we'll come on to the other word that sounds very similar later, which is the petiole, which is the stalk for the leaf. So pedicel, petiole, you can see how there could be some confusion there. And obviously, if you're reading through a bit of descriptive text and it's getting you to measure the length of the pedicel and you can't remember whether that's the leaf stalk or the flower stalk, you're going to be in a bit of trouble. So pedicel is the one for the flower. And essentially that stalk then comes up to the top and usually gets wider at the top, and sort of swell slightly, which is forming the base of where all those floral organs are sort of attached or sit atop. And that sort of swelling area, whether it's just sort of a cup shape for the organs to sit on, or they're all sort of half involved in it, that is known as the receptacle. So essentially that, if you look that up, it just says it's the expanded part at the end of the flower stalk, which the organs of the flower are inserted. So we'll look at some examples of that. Now you can see where the stalk is coming up here and swelling slightly before it turns into the other floral parts. And when you cut that one in half, you can see that we've got sort of the inner workings of the floral sort of reproductive system actually embedded in that. Sometimes it's the receptacle's more just a sort of flat cup almost, that everything just sits atop rather than sort of having it in between. But this basically this cup shape, this bit that is swelling at the top of the stem, is known as the receptacle. And that can be quite useful for some species, where, for example, it's talking about how it is particularly swollen or swollen beneath that. And also we'll come on to exactly how the ovary sits within it or atop it, and can be really useful in the flower key, but we'll come on to that separately. Right, moving on then to the next sort of floral whirl or row of things that come up above that, and that is the sepal or the sepals. And that's basically the outermost row, or referred to as a whorl, of floral organs, and they're usually green, and the purpose of them is to protect the flower when it is in bud. So if you imagine a bud, it's usually, green with those sort of sepals cupping and around it. And essentially those then unfurl and support the petals underneath, and those are the sepals. However, they then throw in this other word, calyx, and basically the calyx is sort of the plural term for sepals. If rather than keep saying sepals, they might just refer in the book as the calyx or the calyces. And that's often because a lot of species might have the sepals fused together, either partly at the bottom or all the way up to form a sort of tube. And in that case, it will be referred to as the calyx. So if you see calyx, it's referring to the sepals, essentially. That is the same thing, just a slightly different way of talking about it. So let's look at some examples. So as I mentioned, you can have them completely separate from each other. So you can see on this lovely crane's bill, the sepals are quite clearly separate, just as the petals are. And they're there just sort of supporting underneath. But you can see on the one behind that they are closed together, protecting that flower in bud. And those are basically free sepals, whereas you can get some that are, as I mentioned, partly fused. So, for example, this one on the ragged robin, we've got this kind of ridged unit that then splits into these triangle shapes, which obviously everyone's more familiar with that as being the sepal, but the whole thing is all made up of that sepal-like material. So that would be referred to as a calyx or a calyx tube that then has these triangular teeth at the end. So that whole thing is the calyx. Something like this campanula, bellflower, you can see again the bud is nicely protected in the sepals, but it's sort of partly fused towards the bottom. So it's still forming a calyx, but it is sort of splitting out into more sort of characteristic like sepals. And then here on this dead nettle family, you can see really, really important for this family, actually, the calyx. So the sepals are definitely fused to form that more calyx-like tube. And identifying kind of how that structure is formed is really important in this whole family. So essentially, we can see that we've got those three sort of pronged teeth at the top, and then a sort of lower two longer teeth at the bottom, if you can make that out. And that would be basically sort of describing almost an upper and a lower lip of the calyx. And because they're not all equal sizes all the way round. And that sort of thing can be really, really important when it's describing it in the little information box, but also in the key when you're kind of going one way or another. So really good to get familiar on seeing different examples of this. Whereas on this pea family flower, you can see also we've got a kind of calyx tube that then goes out onto these long extendable bits here. So again, might be quite useful for identification purposes. And then we're going into a little bit more familiar territory now with petals. And most people, I would say, are probably familiar with what petals are. And they usually the petal would be referring to an individual, but obviously petals, plural, usually a free unit of the second floral row or whorl. And these are the same as sepals. They can be referred to as another name when talking about either multiples or when they are fused together, and that can be known as the corolla. So again, if you're reading in your book and it talks about the corolla or the corolla tube is blue, then now you know that actually it's referring to that whole petal unit, or especially if it's fused together, and it's referring to it as a corolla tube. And obviously they can be really, really important for ID because first of all, they're usually going to be telling you the colour, and also, you know, whether they're sort of shiny, whether they're notched, that sort of thing. And the petals are really interesting in terms of kind of looking at it in the anatomy, because they themselves might be what secretes the nectar at the base or in sort of special spurs at the back of the flower. And they also might have the sort of scent producing glands at their bases, which is what makes the flower smell nice, or bad, depending on what your opinion is. Whereas in wind pollinated flowers, so like the grasses, for example, we don't really have petals because we have no need for them. So they're either absent completely or just so inconspicuous. And there are, you know, non-grass like flowers that have almost inconspicuous petals because, again, they're not relying on attracting an insect in. So we've got the classic kind of petals that are nice and free and separate, and you could pluck each one off individually. Same sort of arrangement for this buttercup, and you might think, oh, somebody's either pulled one of the petals off or one of them is slightly malformed, and not kind of looking great. But actually, this is kind of a characteristic feature of this particular species of buttercup, known as the Goldilocks buttercup, a common sort of one in nice woodlands, really good ancient woodlands and things. And one of the characteristic features is the fact that its petals are often uneven sizes, or you know, some are completely reduced to hardly anything, which I just think is a really interesting feature. Not sure why it would have developed like that evolutionary wise, but very interesting to think about nonetheless. So again, you know, petals playing an important ID feature there. On this barren strawberry, though, it's the fact that they're quite spaced apart enough to be able to see the sepals in between, and that's how spaced apart they are. But also the fact that they're slightly notched, giving us that nice kind of heart shape. So that's a key feature there. This lesser stitchwork, the petals, again, really interesting here. You might be thinking, oh gosh, there's lots of petals, and you might be counting and thinking that there's 10 petals, when actually there's only 5, because each petal is split almost down to the bottom, and it would be called bifurcated, probably in the description, or notched nearly for its entire length, basically. But that whole thing is one individual petal. So really useful to get kind of used to these different features and how they can appear. But equally, we can have non-separate petals, and we can have ones that, just like the calyx, that are fused to create different shapes. So in this lovely harebell, we've got all the petals have been fused nearly all the way to the top to form this lovely classic bell shape. And that is definitely when it would be mentioning, talking about the corolla rather than petals in the description of this species. And then while we're talking about flowers, and at this point talking about whether we've got free petals or fused sort of to a corolla, I thought it was a good time to mention these other words that can be mentioned in the book. So whether we've got a regular or an irregular flower. And we can get even more scientific, and some of the books might say both, or it might just say one. We can either have regular or actinomorphic, or irregular or zygomorphic, which sound absolutely crazy and great. I love the words. But essentially what that is actually saying is it's saying we got one flower like this, or we got a flower like this. And so the regular or the actinomorphic flower will have multiple planes of symmetry. So where I've drawn these yellow lines across is essentially where, if you put a mirror at that point, you would get a complete mirror image that matched it on the other side, that would make it still look like the same flower. And we've got lots and lots of places where we could put a mirror and get that exact image back. Whereas on the other side, the irregular or the zygomorphic flower, you actually only have one plane of symmetry. You could put a mirror down the full central line there from top to bottom, and you would get the nice image back, and it would look like the flower. However, if you did that in the middle, it's not going to give you an image of what the flower looks like anymore, because it doesn't have any more multiple planes of symmetry. And that can be really useful if you're ever going through the very beginning of one of the sort of general wildflower keys. That can be one of the sort of top questions that it might ask you: is it a regular or an irregular flower? And if you don't know what that means, then you've already come a bit unstuck. So now we're moving kind of more into the center of the flower, and we're going to look at those sort of funny little bits that come in all shapes and sizes, and multiple numbers and things like that. And the first part we're looking at is the stamen, and these are the male organs of the flower, and it consists of individual parts that we're going to go through individually, of the sort of the filament and the anthers. And they essentially, at the end, the anthers, they have the pollen. Now, one of the ways that I find really useful for remembering that the stamen is the male part is the fact that it has the word men in it, stamen. So if you can help to remember that that's the male part, and people often find it easy to remember that pollen is the male part, then the stamen is the bit with the pollen, and therefore you can identify it in the flower you're looking at. But essentially, stamen can be really useful in terms of identification. So looking at the number of stamen and counting them, not on a species like this, perhaps, this probably in the description for this bramble would just say multiple or lots and lots of stamen. You're not going to be asked to be counting the number of these ones, but some species might only have two when you think of like the speedwells or something. And so, yeah, the number of stamen can be really useful. But it's also worth remembering that this is an individual flower with multiple stamen. However, you might be forgiven for thinking that this is a flower with multiple stamen, and so you'll be saying, well, this has got lots. But actually, this flower, the devil's bit scabious, is actually multiple flowers on the end of this stalk. So each one of these little purple cup-shaped things, and these ones haven't opened yet, they are an individual flower. So actually, if you were counting the number of stamen per flower, it might be more in the sort of, I can't remember, 2 or 3 stamen per flower. The same with this hoary plantain. You're looking at this thinking, gosh, that flower's got loads of stamen, but actually there's multiple flowers on top of that flower stalk. So each one might only have 1 or 2 stamen. So it's just worth keeping that in mind. That's the stamen. So as I mentioned, we're breaking then the stamen down into its component parts, and you might think that this isn't as important, and generally there's a few species where you have to kind of look at it in this detail, not that many, but it's still useful to know when it's describing them and what these words actually are. So filament is essentially that stalk-like structure that has the anther at the top. Now the filament itself can be relevant. For example, there are some species where you actually have to compare the size of the filament to, different species, and that could be the difference between them. Not very many like that, however. Just so you can see it in a real plant, in this Viper's bugloss, you can see the pink stalks are essentially the filaments there. The anthers at the end are the bits that actually have the pollen in them, and these can be really, really useful for ID. So they are basically the little blobby bits at the end, you can see there. And they are what contain all of the pollen inside. But for example, if you think about bluebells, whether you've got an English bluebell or a Spanish bluebell, one of the first things that can help you, and it won't always help you if you've got the hybrid, but let's talk in simple terms for the sake of this, is that if you've got an English bluebell and you tilt the little bell flower underneath and have a look, the stamen, the anthers should be a lovely creamy yellow colour. If you have the Spanish species, they would be slightly blue tinged. So that's a really easy way next spring, because obviously they're all finished now, to be able to tell whether you have an English bluebell or not. We then get into going away from the male part of the flowers now and looking at the female part of the flower, right in the very centre. It can get a little bit more complicated because there are sometimes other words like pistil and carpel, and then there's gynoecium and things like that. And it's like, whoa, why? How do we have so many words for this one thing? I would say that the gynoecium is the kind of collective term for all of the female parts together. So that includes the ovary, the style, and the stigma. Equally, carpel can be used to refer to that for when you're looking at a simple flower like this that just had the one ovary, the one style, and the one stigma. You could also refer to that as the carpel. However, quite a lot of flowers will have multiple parts, multiple ovary, style, and stigma, in which case you've got multiple carpels. And sometimes you've got that so they're all separate, and sometimes you've got it so that they're fused actually inside a sort of bigger unit, the gynoecium as a whole, and they're all sort of split inside. And we'll get into that in a minute. But essentially, gynoecium, carpel, it's always going to be referring to the whole unit of the female parts. So I was mentioning there about how you can have it sort of arranged. So as in the previous drawing, we had it very simply, one carpel, simple, we've got the ovary, we've got the style, and we've got the stigma at the top. Or you could have three carpels, but they're not fused together. They are separate. And, you know, when you think of buttercups, if you ever, go and look at a buttercup in close up, you've got your lovely yellow petals, and then right in the centre, you've got this kind of jumble of yellowy green sort of blobs that aren't the stamen, and those are the unfused multiple carpels, and they're kind of an early evolutionary plant, so that's why it's kind of quite simple and basic and obvious. So do go out there and have a look at a buttercup the next time you're in your garden or out on a walk. But essentially, they're very, very separate from each other. However, other species might have, or other families might have three carpels, but actually fused together, which means if you cut it very neatly in a cross section, you would see that you've still got three separate chambers of ovaries, and coming out the top, you might have three separate styles and three separate stigmas, or you might just have three separate stigmas coming right out the top. But essentially, those are the carpels that are fused together. So we mentioned that that's the whole unit, the gynoecium or the carpel. And then when you break that down into that individual parts, we've got the ovary at the bottom. Now, this is the ovule bearing part of the female organs, and it's usually basically enlarged to have all of that. And eventually that becomes the fruit once it's fertilized. The ovule is essentially the tiny immature seed in the ovary. And you can see here when we cut one in half, so this is the whole ovary, and when we look at that really close, you can see the little ovules inside. So that is the seeds for the future in there. Now, I wouldn't say that there are that many times that you need to know details about the ovary when it comes to different species. However, the fact that the position of where it is, that is going to be potentially more useful, which we'll cover in a minute. So we've got the ovary at the bottom, then we've got that sort of stalk-like structure again. So in the male part with the stamen, that was called the filament, whereas in the female part, that's called the style. And that is basically the part between the ovary and the stigma at the top. And it's often slender, and then sometimes you don't have it at all. And actually the stigma just sits on top of the ovary. So yeah, you don't always have that. So for example, in this one that I cut open before, you can see we've very clearly got a nice sort of style going there, joining between the two. And that can be very useful for identification purposes. If we take, for example, these two species, so we've got the paler one is the field rose, and the pinkish one there is the dog rose. Ignore the colours, they tend to vary massively between themselves anyway, so you can get pink or white. But what can really help you in identifying the two different species apart is whether you have a style or you don't. So in dog rose, we don't have a style. So you've got the ovary essentially, and the stigma just sits atop it completely. It's sort of a jumble of a mess on the top there. It's not very obvious, but blobby bit that is the stigma. Whereas in the field rose, we've got a very, very clear style separating it out. And that can be a really useful way to tell the difference between the two. And it does persist even into when you've got the hips. you can often see on the field rose hips the little style sticking out of the top. So really useful. And then, as we mentioned at the top, you do have this stigma. So this is essentially the pollen receptor of the female organs, which might be, as I said, directly on top of the ovary or elevated up through the style. And the stigma itself can be quite variable depending on the species. All manner of shapes and sizes a stigma comes in. So, for example, here we've got almost, a sort of 5-branch stigma, and that can be quite useful for knowing with the description of it. This one's got 3 little recurved bits, but essentially lots and lots of variety. Again, can be really useful in identification, and knowing what your stigma is will help you here. So willow herbs, the epileniums, are really, really tricky group to identify. But one of the things that divides sort of one half of the species from another is what shape your stigma is, essentially. So you can see on square-stalked willow herb here, it's what's referred to as a club-shaped stigma. So it's just a simple sort of elongated bit at the end of the style. Whereas on the broad-leaved willow herb, you can see that we've got those 4 sort of divisions, if you will. So yeah, 4-lobed or 4-spiked. It's referred to different ways in different books, but we've essentially got the 4 divisions of the stigma. So again, that would help you when you're looking at the description of the species, but in the key, that could be an absolute separation of going one way to the other. So really useful. And I mentioned before about this, how important it is with the ovary position. And it isn't quite as black and white as whether you've got a superior ovary or an inferior ovary. There is a little bit of some that are called sort of in between, but this is the basis of it is essentially, have you got the flower parts, so your male flower parts and your sepals and your petals coming in underneath where the ovary starts and the stigma starts, or have you got them sitting all on top of the ovary? And that's essentially what it wants you to look at. And again, this is really useful if you are ever keying something out from the very beginning of like a general key. It will get you to sort of look at this. And you can see quite clearly on an example like a poppy, where you've got your obvious ovary sitting right on the top of everything else underneath it. Whereas on something like a rose or this one, you can see that all of that sort of ovary stuff is further underneath the petals and the sepals. So that would be more inferior. But it can be useful. And don't worry if this feels confusing, because this bit can even stump some of the best botanists. So it can be quite a difficult thing to wrap your head around. But I thought, let's not shy away from difficult things, because everyone wants to learn, and it's a good place to start. So we've looked at some of the sort of basic structure of flowers, but obviously they don't all look like that drawing I did, and quite often they're not like that at all. So we've got huge variety in the way that flowers are formed. So we've got what's called a classic pea flower. Again, these would be zygomorphic or irregular because they only have that one plane of symmetry. But the helpful thing about all of the pea family is that's one of the characteristic things about the family is that it has what's called a pea flower. And I know that sounds silly because if you've never seen a pea flower before, you wouldn't know what that means. But essentially, it's quite recognizable with the big sort of banner petal at the back. You've got the wing petals and then the keel petals, and it looks classically like a pea. But all of the flowers in that family have a pea flower. It's just some are much more obvious and big, and some are tiny, like on a clover. They are still pea flowers, but there's just lots and lots of them on one flowering head. So again, another one to get out there and have a look at. Have a look at your white clover, maybe with a hand lens, and just see how they're all individual little pea flowers on the flower head. Then you've got things that are completely not recognizable as a traditional flower at all, so probably wind pollinated in this case, but still have all of those essential flower organs, and sometimes they still might need to look at some of those features for ID. Then you've got the wonderful world of orchids, which are completely different and have, in fact, whole set of their own terminology when it comes to their flowering arrangements, because they're not now nicely divided into petals and sepals. They're sort of adapted sepals and petals all formed into one, and other bits and bobs. So yeah, quite complicated when it comes to orchids. And if you've ever tried identifying some of them when they easily hybridize, it can be really, really tricky. We've then got things like the euphorbias, the spurges, and they basically have also quite a strange flower structure, whereby you've got these half moon shapes are actually glands. You've got the classic male stamen there, and then you've got this strange little ball on a, which is actually the ovary, and then the stigma coming out the top, and then these other little bits are actually bracts hiding more flowers that will be coming out separately. So again, it gets even more complicated. And then at the sort of pinnacle of the sort of complexity when it comes to evolutionary design, I think, is the fact that things like the daisies and the dandelions and all of those families, where you've actually got hundreds of flowers actually on one flower head that have just been sort of arranged slightly differently. So each one of these white petals is actually an individual flower, and each one of these strange little open yellow things is an individual flower. You can see the stamen and the stigma poking out of them. And that's the same for things like the knapweeds and that. And it's because they've basically got two different flower types on them. They've got ones that are like ray florets, they're called. And in the case of the knapweeds, they're more like a tube. And then you've got disc florets. Floret just means flower, but we're talking about it on a sort of small and individual scale as part of a larger flowering head, usually. But compound flowers, really, really interesting. And things like the daisy family, it wants you to look at whether there are additional things in between the disc florets. So you can see on this illustration, we've got these strange little sort of bracty type things that aren't really serving a purpose, and scariest bracts and things like that. And then obviously when it develops into the seed and then the sort of fluffy bit, these are all really, really interesting. So then we're moving away from the flower briefly, just to go through some of the real basics of some other parts of the plant. So have we got, for example, our heads wrapped around some of those funny little words like petiole and stipules? Again, really, really key when it comes to identifying plants, but sometimes it can just get a little bit confusing. Pretty sure most people will know what a leaf or a blade is when it's talking about grasses and the stems. Sometimes it might refer to things about like internode or node, and that is usually just talking about literally the part of the stem where there's nothing going on versus where there's something going on and things are branching off, being the node, and the gaps in between being the internode, and obviously you've got the roots underneath. Then when you're looking at it in a little bit more detail, as I mentioned, we've got that little stalk for the leaf called the petiole. And then sometimes, not all species, not all families, have little extra bits called stipules at the base of where the leaf comes out. And that's very different to the bud of where another leaf is coming out, which can often do a really good job of looking like stipules. But usually stipules are leaf-like, but often very different to the actual leaves. Sometimes not, though. Really good family to go and look at stipules sort of in the flesh, so to speak, is family. So all of them have really interesting shaped stipules. If you've ever looked at meadow vetchling, its actual leaves are sort of formed of long leaflets, but the stipules are almost like arrowhead shapes going up the stem. So it's worth going and having a look at that. So as I mentioned, yeah, the node is just where we've got things branching out, leaves and things like that. Then, oh, thank you, no worries to Rachel there, and I hope you can catch up the rest of it on the video. So yes, and then we've got these stipules, which essentially like these extra things. They're not necessarily red. I've done that so that you can see that they're different, whereas actually we've got a bud in the leaf there. And the petiole is the actual stem, the little stalk of the leaf, as we mentioned. So I did mention that we were just going to do a little test, and obviously being a webinar, I can't kind of hear your results, but if you want to have a go at putting the answers in the chat or just doing it privately to yourself, that's absolutely fine. So see if we can remember the names of what we've just gone through. So essentially, we've got number one up here. Anyone got any ideas to themselves or want to put in the chat? It's absolutely fine. I'll give it just a couple of seconds. Yes, we've got answers already, and yeah, we are correct. Nice one. And then next we're asking about this specific little bit here, this actual stalk-like bit. Anyone got any ideas to themselves or in the chat? Feel free to guess away. Style, absolutely. I haven't done it in necessarily the order that we've gone through everything, but I think that's sometimes quite useful rather than just sort of following the same pattern. But also it was partly to do with how I could label the diagram. So number 3, we're looking at basically the units here that are sort of big and usually coloured and sometimes scented. I think we all know that petal, but that other word that's worth remembering is that corolla. So that's worth, just making a note of that. And then we're talking about basically this swollen bit at the top. Now, we did this at the very beginning. Can anyone remember the name of this one? Again, it's not used hugely when it comes to sort of talking about an ID, but, yeah, perfect. People have got it. I'm pleased. And then the next one, number 5, we're talking about this actual separate part of it, the swollen bit at the base. It might have little tiny forming things inside it. Yeah, everyone's got that. Nice one. Now we're talking about the things that are sometimes leafy underneath the petals that keep it nicely protected in bud. And I think it's worth saying, there's one word we're more familiar with than the other. So if anyone could not say the other thing that might be talking about it, whether it's fused or, in the multiples. So beginning with C, anyone remember that? Yes, nice one. Sepal or calyx, very good. And then we're talking about this whole unit here. And again, there are kind of 2 different words that we could use for the next bit, whether we're talking about it sort of on a larger multiples or just a single individual, which is the gynoecium or the carpel. So yeah, bit of a weird one that. But again, mostly the ID books are looking at the individual parts, so the style and the stigma anyway. Oh, apologies, I've gone ahead before, but that's the filament is the stalk-like bit for this, but what is the name of this whole unit here, the male part, that includes the filament and the bit at the top? Yeah, excellent, stamen. And then finally, what is the name of the bit that contains the pollen at the top? Yeah, perfect, nice one. And then we've only just done this, so hopefully everyone will remember, but can anyone remember the name of the part of the stem where something's happening, like branching out, or there's a stalk coming out? Perfect. Node. And then the bit in between where not a lot's happening. Anyone? Nice one. I'm loving how quick you all are, by the way. This is brilliant. And then over here, can anyone remember the name of the parts that are not leaves, but they're there at the base sometimes, but not in every group? In fact, some families are, definitely have stipules, some definitely don't. The rose family pretty much all have stipules. Oh, I just realised I've given it away. But there you go, you'd already answered for me anyway. And then finally, that funny little word that's referring to the stalk of the leaf that could be quite useful. Petiole, that's our word we were looking for. So I'm going to do a little bit of time for some questions, but I thought I would cover off some of the ones that I get asked most often, which is what are the best books for plant ID if you don't already have any? So I would say always a really good one for beginners is the Frances Rose Wildflower Key. It's an excellent book. Got several copies of it here. And it's a really good solid starter book. And the descriptions are fantastic. Anything that's kind of in bold usually means it's like really important when it comes to identifying that species. But the only thing is it doesn't have grasses, sedges, rushes, ferns, that sort of thing. So it is very much more just for the flowers. Whereas if you're then wanting to move up from rose, or you want to start with all of them in one book, then this is my absolute go-to. It's my personal favourite that I take out every time I go in the field, and it's the Collins Guide. Really important that you get the second edition here, because the first edition has a few little errors with some of the key. So this is a really, really good book. Really recommend it. It is heavy. It's a bit of a brick, but I guess because we've got the grasses, sedges, the rushes and the ferns in it, it is a little bit more chunky. So, are all flower keys the same? Is sometimes a thing that you might be wondering. So, we talk about flower keys, and essentially they're pretty much all the same in terms of how they are done. They're normally all couplet options. So, you start with one and then you look at the two options, and essentially you choose, make a choice out of which of those options you've got. And then you see whether that then takes you to the next couplet or further on in the key. And most of them are all like that. And they do generally all follow the same sort of questions about the plants. Obviously, this is a general key. So this is starting with basically you've got a plant, but you absolutely don't know what family it's in or it's anything like that. Whereas most of them also have family keys within them, which is really useful. So if you know that you've got a buttercup, but you don't know which one it is, you can start in the buttercup key and it'll ask you just, questions that are just going to be related to buttercups rather than to whether it's a tree or a grass or whatever. So what happens if you can't remember all the terminology we've covered or it mentions words that we haven't seen? Then always look at the glossary at the back. It's always really useful to just have a quick look there and just remind yourself, oh yeah, so what's the petiole again? I can't remember whether it's the pedicel or the petiole, but it'll say in the glossary, so it's really worth that. In fact, I've got a separate book called the Plant Glossary, which goes into loads more details and has lovely little images to talk about all of the different parts of the anatomy. So really useful book there. Now, plant apps. So definitely are getting better and better by the day. My personal favourite is the Flora Incognita. And I actually do definitely use it myself, usually to confirm kind of what suspicions I have, but I particularly use it when I go abroad because I'm often completely none the wiser about what I'm finding. But obviously, if you're very new and you're a beginner, then do have a word of warning that don't rely on them essentially because and they do give you a percentage of how accurate they think they are on the guess, but sometimes they're not always taking into account the particular place you're in, and some of the sort of subtleties of what you're looking at. Some species are much easier to tell than others, but if you've got no idea of where to start looking in your book, then an app could actually help you kind of lock down maybe the family of where you need to start with. So hopefully that's useful. So just covering what we've learned again, we've done the receptacle and we talked about that difference in ovary position, whether it's a superior or whether it's an inferior. And again, I mentioned about how difficult it is. So don't worry if that's not something that's completely obvious to you when you look at a flower, but it could be worth going out there and having a look. Again, the buttercups, obviously that's going to be a superior ovary because all those carpels are sitting all together on the top. So yeah, that's one useful to look at. And the rose, as I say, it's sort of halfway between the two. We've got the sepals and the calyx there, then protecting the bud, and they're supporting the flower afterwards. We've got the petals or the corolla. We've got the carpel, gynecium, ovary, style, and stigma, sort of all together at the female unit. And then we've got the stamen, which is made up of the filament and the anther. And then we've talked about leaves and the nodes and the internode and the roots and the petiole, the stipules if they're there. And again, that's just the node. So I think we've probably got a good 10 minutes or so for any questions, if anyone would like to share any questions.
That's brilliant. Thank you so much, Sarah. That was amazing. While anybody wants to type in any questions that they might have, either in the chat or the Q&A, I just thought I would do a quick shout out to another webinar that Sarah is putting on for us. I'm trying to think now, the date of that webinar is a little bit later this month. I believe it's the 23rd of June. And Sarah's going to be looking in greater depth at grasses and looking at our usual suspects. So I just thought I would mention that while giving anybody a chance to write in their messages and questions. Also, just a note to say, we've mentioned a couple of times, and you've probably noticed that this session is being recorded. So yes, don't panic if you forget anything later or you want to come back for a refresher. I will be popping it on the NPMS support YouTube channel. In due course. I just need to download it and re-upload it. Right, that was the quick 2 plugs that I just had to put in there. There we go. I can see we've got 2 questions thus far. One
Yes.
regarding on the Q&A, Sarah, do you think OBS Identify is a good app to use as a starter for where to look? Any thoughts on
It's not
identifying?
one I've never used it, so I couldn't comment on whether that one is a good app to use. I mean, my particular favorite is still Flora Incognita, and I'd say even as a beginner, that one is pretty easy to use. It's free. It's also citizen science, which we're all fans of, let's face it. So yeah, I mean, I don't work for them, by the way, so I'm not plugging them for that reason. But it's just I've never used the other one, so when I'm out with my colleagues, we sometimes try different apps to see which ones, and I just find that Flora Incognita always gets better than everyone else. One thing I would say as a little tip for using it is that sometimes, and it might just be my phone, that when it asks you to take a photo when you've got the app open, it's sometimes not very good at getting a clear shot. So sometimes I take the photos first of the different parts of the plant, and then when I'm using the app, I then upload an existing photo, and that seems to work better. So yeah.
That's a really good tip. Another thing you could do as well is put a piece of paper or something behind the photo, behind the plant to get a better, photo in focus. Another really lovely thing of having you all here is that while we don't have experience of Obs Identify, somebody here has in the
Yeah.
chat. There we go. Somebody has used it and they find it as good as PlantNet and often use both to cross check if I'm not sure. Brilliant, brilliant tip. And yeah, I'm really glad that Sarah said as well, actually, they're a really good starting point, but always have that little sort of thought in the back of your mind just to take everything with, they're fantastic, but we tend to say to use them as guides and
Yeah.
suggestions rather than yes, the be all and end
And
all
the other
answer.
thing to be wary of is that this try and double check the scientific name, because often they have completely different common names than we're used to, and often more Americanized. And so you could be put off by thinking, well, it can't, well, what's that? But if you check the scientific name, it should be able to match. Interestingly, we did put them through their paces a little bit the other week when we were running a mock FISC. For anyone who doesn't know what a FISC is, it's essentially a botanical exam. Yes, it is quite scary. And we tested the apps on these species to see how it would do if it took a botanical exam, and it was very, very good. Let's just say it got 100% in the first 10 species that are supposed to be the easier ones, and it would have got sort of 75% in the next part of the test. So yeah, very,
It's
very, they are
encouraging,
getting
isn't it?
better. Yeah.
Well, I think, am I right in saying the more people use them, the more that add to those databases, the better they're going to get,
Yeah.
essentially,
Yeah,
because the
because
more information
you're
that's being
training
gathered.
the AI.
Yeah,
Yeah.
yeah, indeed. We've
Uh,
got another couple of questions in the QA. Let's have a look. Oh, one regarding do all plant apps feed into county records? That's a really good question.
so with the Flora Incognita, it doesn't feed into the county recorders at all, but it does go into their own citizen science project. So I would say I would use them more for just ID rather than for records. However, if you're using iRecord, then they do get filtered through to the county recorder, but sometimes it depends on the county you're talking about, but sometimes they can be quite slow to get the results. There is obviously a BSBI app now. I say app loosely, really, because it's not something you can download off the sort of app store. It's more a sort of pinned browser site, but you can record ad hoc plant recordings or surveys in that, and obviously that will go more directly to the county recorder. So yeah.
Brilliant, thank you. And we've got one more question here regarding identification level for surveying. So someone's asking if I'm able to identify, say, 99% of species in a square, would that then be inventory level? If I take the wildflower level, am I just recording the wildflower species selected for the habitat in the species? Yeah, that's exactly right. If you are recording at the wildflower level, then you do only need to record those species that are within the species list of that particular habitat for that plot.
With the little black flower
Yep, with
symbol.
a little black flower symbol, that's right. If you're then recording at the indicator level, that's sort of it is an expanded list of species. So it jumps up from, say, 10 to 15 species up to around 30 species per habitat type within your plot. So you really are only recording those species. Of course, there'll be other species there within your plots, or not all of those indicator species will be there, but they are the ones that we're interested in finding out for that habitat, what's present, what's absent. And then again, at the inventory level, that really is for folk that are comfortable identifying all vascular plant species. Um, there are some assemblages in there, but yes, that's for people who can identify absolutely everything. And we do say, unless you are going to record all vascular plant species, to please select indicator level. Otherwise, if we select inventory level, for example, but there's some things that we can't quite identify, so we don't note them down, that then assumes that they're absent rather than they're there and we just couldn't ID them. So unless
Yeah.
you're absolutely confident, recording and identifying absolutely everything in your plot, please do record at the indicator or wildflower level. Hope
Yeah,
that's
I mean, I would
clear.
say if this person says that they can do 99% of the species, I'd say you're pretty much nearly there, but
Yeah,
maybe go at indicator level, so that's above wildflower, and see how you get on. And you can always, I think there's options to write additional species in comments and things like that. So yeah,
yeah.
maybe.
That's absolutely right. Not just in comments, but you can actually record extra species
Yeah,
as well. So along with our the 3 recording forms, you always also have an extra species recording form, and that's really to record anything at all that you've observed and you can confidently identify, but aren't on the indicator wildflower list for that habitat type. So it's all
yeah.
useful information. It's just not used in quite the same way.
Great.
So that's done. Right, my golly. Any more questions coming in? I cannot see any more questions coming in at the moment.
No, I don't think so.
So I'm going to have a chance again to say an email has gone out to all volunteers, NPMS volunteers today, with some information about our lovely events
I
and
saw that.
activities we've got coming up over the course of the season. If anybody happens to be around Bristol or Bristol Festival of Nature this weekend, please do pop by to the NPMS stand to say hello. And yes, do take a look at those, both the training events on our new training events page, and also take a look at the webinars. And they're housed slightly separately because, of course, our webinars like this one are open. You register and you head straight to Zoom to be able to access those free webinars to all. But the training events page is solely for registered NPMS volunteers. So do
And
take a
I
look
have
at
to
the
say,
events
I
page.
thought the email showed some great training coming up in the field and everything. So yeah,
Oh,
really good.
yes, we're
Um,
looking forward to this season. Um, Brilliant. And it's also worth noting that throughout this season, we're celebrating our 10-year anniversary, which is very
exciting.
exciting. So we're all super
Yeah.
keen. Right. Let's have a look. Any more questions popped up in chat there, Sarah? Let me see.
I don't think so. I think we're all good.
Nope, that's
Hopefully that
perfect.
means it was clear.
Yes, that hopefully that means it's clear. Yes, and brilliant. So everybody, thank you so much again for joining us and taking time out of your lunch. I hope you managed to quickly grab a sandwich or something now. And we'll be seeing you soon. Thank you
Yeah.
so much,
Thanks,
Sarah. That was
everyone.
fab, as
You're
always. Thanks a
welcome.
lot. Bye now. Bye.
Thanks. Bye. Bye.